is Hannah Doherty. Hannah is a second-year PhD student at the University of Birmingham on the Wellcome Trust AAMR programme. She's a member of Dr Manuel Banzhaf's group, and her study of interest is on chemical genomic screening with Pseudomonas aeruginosa, with the aim of finding potential solutions to antimicrobial resistance within Pseudomonas aeruginosa. So over to you. Thank you.
Hi, yeah, so I'll be talking about high throughput approaches to chemotype and phenotype Pseudomonas aeruginosa and other bacteria. So for this project, the bacteria that we phenotyped was Pseudomonas aeruginosa, and published single gene knockout libraries of two strains we used, PA01 and PA14. The PA01 dataset included 5,570 single gene knockout mutants, and the PA14 set contained 5,814 single gene knockout mutants. But there's actually a crossover of 5,336 genes. So interestingly, since they are both genetically very similar, but phenotypically very different, by using both libraries, we hope to find clues on how this phenotypic diversity is genetically encoded. So to analyze the Pseudomonas libraries, we used a high throughput approach called chemical genomics, which is a means of evaluating the phenotype of single gene mutant libraries within a variety of chemical stresses and conditions. So the first stage of this is making growth plates. So these are a series of solid media plates supplemented with different conditions. For example, antibiotics, stresses, human-targeted drugs, mimicking of the host environment, differential medias, and other things as well. And for this study, we tested around 200 different conditions. So then a pinning robot is used to pin the mutants onto plates from 96 well source plates to 1,536 colony plates, as shown here, where each colony represents a different mutant. And biological replicates of each plate were produced using different source plates. So then once we've pinned, we leave them to grow, and then we image the plate, and then we use IRIS, which is an image analysis software, to analyze the phenotypes. So this can detect different phenotypes such as size, circularity, biofilm formation, and as well as colony integral opacity, which is a 3D measure of colony size, which takes into account both the height and the area of the colony. So it's a slightly better means of evaluating colony size. So once we have the size data for each plate, the data set then undergoes quality control steps to ensure that only the best data is actually going into the data set. And for this, we use ChemGap, which is a software that I am currently producing for the analysis of chemical genomic screen data. And then once the data set is cleaned, we can then give the colony sizes S scores, which are a measure of fitness. And then these scores can be clustered and displayed in a heat map for the discovery of hits within the data set. So one of the important stages of the pipeline is the quality control steps, which are implemented by ChemGap before scoring, since to get good data out, you want to put good data in. So the first thing that we do is that each plate within the data set is normalized. This is done in two stages. The first stage, the outer edges of each plate are scaled, such that the median of the row or column that the colony is in is scaled to the plate medial mean. Thus, the outer edges are scaled to the center of the plate. This is because the outer edges tend to be larger, since the colonies have less neighbors, so they don't compete as much for resources, and so they can grow more. And then the second stage, we scale the entire plate, and we scale it such that the whole plate, the middle mean, the plate middle mean, is equal to the median size of all colonies within the study, not just from the plate. And this normalizes such that all plates have a similar distribution and mean peak, since some stresses may result in worse growth than others. Therefore, all plates can be comparable, as you can see here on the diagram on the right. So once the data set has been normalized, we put it through a series of statistical tests to evaluate which plates or conditions might be detrimental to the overall data set. First, a Z-score test is performed between each replicate of a mutant within the same condition. This highlights which colonies are outliers and classifies them as either smaller or bigger than the mean, designating them as S or B. And then normal colonies are designated in N. Zeros, where not all colonies are 0, are designated Z, and where they are all zeros, TZ for true 0. The percentage of each of these for each condition and for each replicate is then calculated. So then the next test that we use is the Mann-Whitney test. And this test computes if two sets of data fall into the same distribution. So if replicates are reproducible, then they should have the same distribution, as you can see here with A, B, and C, for example. However, if one replicate differs, such as D, this is flagged up by the test, and we can know which ones are problematic replicates. So each replicate is compared to each other, and then the mean p-value is taken to represent each replicate. So the smaller the p-value, the less likely they are to be of the same distribution. So then the variance of these mean p-values is also then computed, and conditions with a large variance likely have very different distributions and are therefore not reproducible and damageable to the dataset. Finally, a variance analysis is performed, and the average variance of replicates for each condition is calculated. So once these tests have been performed, the number of conditions with a certain percentages of plates lost is calculated at different thresholds for different tests. For example, if the count of conditions with different percentages of plates lost for a normality threshold of 20%, where if a condition plate has less than 20% normal colonies, then it would fail and the plate would be lost. So based on this, we can choose thresholds which give a good balance between removing detrimental plates or conditions and keeping enough data that the data set remains informative. So the conditions and plates which fail at the chosen threshold are then removed from the data set. So once the data set's been cleaned, then we can score the colony sizes. And for this, we use S-scores. And S-scores are a measure of fitness based on how far the colony size deviates from a simulated wild type, which is the median of the mutant of interest across all conditions. Since most gene mutations won't be essential in most conditions, the majority of colonies will have an S-score close to 0. Therefore, S-scores less than -3 or greater than 2 are considered significant. So now that the data set is scored, we can try and look for hits. And the way that we find hits is based on the principle that genes that are involved in the same function are likely to respond similarly in the different stresses. For example, as with a car, if you remove the wheels, then the car won't drive. Also, if you remove the engine, then the car won't drive. It doesn't matter what you remove, they're both essential for motility, so they're going to result in the same effect. The same is often true for proteins and complexes. If you remove one part of a protein complex, the whole thing is unlikely to function, and therefore it doesn't matter what piece you remove, a similar result will come out. So this translates within the S-scores and the phenotypic profile of the mutant. The phenotypic profile is the phenotype of the mutant across all conditions. So if we have 4 mutants that are all responsible for a different section of a protein complex, then it's likely that they will have a similar phenotypic profile, as you can see here. Because of this, when you cluster the data, they will cluster together. This is important because it allows us to find the functions of orphan genes. For example, if we cluster the data set and we have 4 known genes and a mutant X, and if mutant X clusters with these 4 known genes, then it's likely to be involved in this protein complex, and we can therefore assign a function for mutant X. So on this principle, we cluster the entire data set and we get something that looks like this. So the colors correspond to S-scores, and we look out for clusters of green or red, as these are the significant scores. So this gives us an example of functional clusters and project ideas that we can then follow up on. For example, one functional cluster that came up in the dataset was MEX-AB OPR-M. And this is an efflux bond. Obviously, this has already been discovered, but by testing different compounds, we can see which compounds are the substrates of the pump. So mutants within the pump will become sensitive to the same compounds or antibiotics, such as beta-lactams, aminoglycosides, or quinolones. It's also quite important to note here that since we know these genes form part of a protein complex, it's very promising that actually they do cluster together and they do show up in our data set as clustering together, because this kind of provides evidence that actually the method is reliable for identifying and clustering genes with similar functions. So another hit that we found was these two genes, PA2403 and PA2404. These both clustered together and they both had sensitivity to adult bovine serum, cobalt-2 chloride hexahydrate and copper chloride hexahydrate. And basically, these are metal stress conditions. So we can assume or hypothesize that perhaps these two genes are involved in metal stress. So these are just two snippets of hits that this method can elucidate. But the use of chemical genomics is a very powerful method of finding functions for orphan genes on a genome-wide level, including antimicrobial resistance genes and potential drug targets. Because of this, we also aim to test in other bacteria, including Acinetobacter baumannii, Klebsiella pneumoniae, and Mycobacterium bovis BCG. We have the mutant libraries for all of these strains, and so we're open to collaborations with people who have interest in these species. Just contact me or my supervisor. So thank you for listening, and thank you to my supervisors, Dr. Manuel Banzhaf and Dr. Dinesh Muradi Garaband. Thank you very much.